Harry Compton Crick was born on the 8th of June 1916. He studied physics at University College London, and he then changed from physics to biology and began to work at Cambridge University.
James Dewey Watson was born on the 6th of April 1928 in Chicago. He studied at the universities of Chicago, Indiana, and Copenhagen. In 1951 he moved to Cambridge University and met Harry Crick.
Watson, Crick, and another scientist called Maurice Wilkins shared the Nobel Prize for Medicine in 1962 for discovering the structure of DNA. Rosalind Franklin, however, did not receive the Nobel Prize, despite her key experimental work, as she died in 1958, and the prize cannot be awarded after death.
What Watson and Crick discovered was the structure of DNA and not DNA itself. They showed that it was arranged in a helical shape made of 4 bases: adenine, cytosine, thymine, and guanine.
DNA is the blueprint for all multicellular life on Earth, and it determines everything about you, from your hair colour to your height, and all the information for it is carried in the 4 bases. In the human genome itself, there is enough information to take up 30 megabytes.
Watson and Crick solved this using stick and ball models, but they actually came up with the idea when they saw one of Rosalind Franklin's experiments without her permission, effectively stealing it from her.
The discovery of the structure of DNA was one of the last pieces of the puzzle. Gregor Mendel had discovered that traits could be passed from parent to offspring several years earlier with his pea plants. Also, DNA itself had actually been discovered in the 1800s by a Swiss physician called Friedrich Miescher. He discovered a substance in the middle of cells which he called nuclein. He had actually discovered DNA.
With the discovery of the structure of DNA, they could not only tell where the genetic information is stored, but also how DNA replicates. It splits down the middle and becomes 2 single strands. These single strands serve as templates for building 2 new double-stranded DNA molecules. The discovery of DNA structure divided the world of molecular biology into before and after the discovery of the double helix. The discovery of DNA created an entirely new branch of biology called genetic engineering. This has allowed us to create retroviruses to combat diseases such as AIDS and to combat cancer. The entire human genome has been mapped out, and if you send a sample of your own DNA off to a lab, they can now tell you where your family originally came from. We can now map the genome of animals that died hundreds of years ago, and we could possibly bring them back. By manipulating the genetic structure of plants, we can make them resistant to insects or diseases, make them use up so little water that they could thrive in the Sahara and produce huge crops. These advances have, however, created bioethics, which worries about the impact of biotechnologies on the environment, human rights, and dignity, and whether we should even alter the genomes of organisms, because we are then playing God and we have no idea what the consequences would be. If we are already disrupting ecosystems by putting genes into plants, what would happen if the whole ecosystem breaks down because of one of our manipulations?